Did you know that inside every single cell of your body there's a 3 billion letter instruction manual? This is your DNA, the code of life. It's the blueprint that builds every organ, every protein, and every feature that makes you who you are. Every one of us is born with around 5 to 7 defective genes. These are mutations we carry from the moment of conception, and yet most of us grow, live, and stay healthy. How is that possible? The answer lies in redundancy. The human genome is incredibly resilient because almost every gene exists in 2 copies, one from your mother and one from your father. So even if one copy is faulty, the other one usually steps in to do the job. This built-in backup system is a lifesaver, allowing us to survive despite carrying genetic flaws. Trouble only begins when mutations hit both copies of a crucial gene. That's when biology can take a very different direction. So what exactly are these mutations? A mutation is simply a change in the DNA sequence. It could be a single letter swap, a missing piece, or even a rearranged chromosome. Some mutations are passed down from our parents, called germline mutations. Others accumulate quietly throughout our lives, caused by things like oxidation inside our cells, radiation from the outside world, or just simple errors when our cells divide. But here's the fascinating part: not all mutations are harmful. In fact, mutations are the very reason you and I exist today. Mutation is the raw material of evolution. Every new trait in human history, from eye color to immune strength to the ability to digest milk, began as a random mutation. Most of them do nothing. Some are harmful. But a few rare changes give an organism a tiny advantage. Over thousands of generations, these helpful mutations spread, allowing species to adapt to their changing environment. Without mutation, humans would never have adapted, and we might not have even evolved into what we are today. We wouldn't be able to resist infections. Mutation isn't a biological accident. It's the engine of life itself, but evolution is slow. Inside your body, that same mutation process is happening every single day, and sometimes it goes wrong. Your genome contains powerful genes that control growth, repair DNA, and tell cells when to stop dividing. These are called tumor suppressor genes, and they act like the brakes on a car. When mutations hit both copies of these critical genes, the consequences can be catastrophic. Let's look at a few of these cellular guardians. The TP53 gene is one of the most important we have. It's often called the master guardian of the genome. When both copies are damaged, it can lead to a wide range of cancers, including breast, lung, and sarcomas, as well as a hereditary condition called Li-Fraumeni syndrome. Then there's the RB1 gene, which controls a key checkpoint in cell division. When children are born with both copies of this gene lost, they develop retinoblastoma, a deadly tumor in the eye's retina. If only one copy is missing from birth, there's a lifelong risk of losing the second one, which can lead to bone cancer. The APC gene is another critical player that regulates how cells stick together and divide. Mutations here are the hallmark of a condition that leads to early colorectal cancer. You can think of cancer as evolution happening inside a single cell, uncontrolled, accelerated, and incredibly dangerous. A normal cell doesn't just become cancerous overnight. It needs to acquire multiple mutations through what's called the multi-hit model. First, a mutation that pushes the accelerator for growth. Then, another that disables the brakes. Maybe another that helps it hide from the immune system. This is why cancer often takes years or even decades to develop. The great irony is that the very same process that allowed humans to evolve is the one that allows cancer to evolve inside us. Mutation is necessary for life, but when it becomes excessive or uncontrolled, life starts to work against itself. The good news is that we can now use this knowledge to fight back. Today, we can sequence a tumor's entire genome and read its mutations like a roadmap. This has led to the creation of incredible targeted drugs, medicines like EGFR inhibitors, ALK inhibitors, and PARP inhibitors. Each one is designed to attack a specific mutation driving the cancer. By understanding the mutations, we understand the cancer, and by targeting them, we are changing its future. Thank you for watching. This is Dr. Danish. Subscribe for more deep dive medical explainers.